Dear students, the topic of today's class is free radicals, antioxidants and oxidative stress. Over the years, free radical biochemistry has been studied by scientists all over the world since Dr. Halliwell, 1980 around, discovered the etiopathogenesis of free radicals and its association with different disease. However, nowadays, not only the disease are associated with this free radical mediated oxidative stress, and different antioxidants are now studied for the treatment of different disease. So oxidative stress is playing a key role in many of the cases. You have to get the basic ideas of this particular topic. So we are going to see what are free radicals, what are oxidants, what are antioxidants, and how oxidative stress occurred in our cells. Next slide, please. So first we are going to learn what are free radicals. What are antioxidants? So free radicals is a particular species, and oxidant is the group of that species which are reactive, and free radicals specifically are assigned to be the more reactive molecules. So whether the free radicals are always the bad guys, what is your concept? You have to develop this concept. What are biomolecules? What biomolecules are antioxidants? Are they supposed to protect always? So whether these antioxidants are our little heroes, let's develop our concept. Next, please. By definition, free radicals are atoms or molecules with at least one unpaired or unshared electron, causing it to be more reactive. As I've already told you, there are different reactive molecules, reactive species developed in our body, in the cells, out of which one are reactive oxygen species and one are reactive nitrogen species. Now, out of the reactive oxygen or nitrogen species, some of the molecules are highly reactive because they have this particular criteria, or one electron is unshared or unpaired. In this group of oxidants here, or oxygen is definitely a reactive molecule, it's oxidized. Now, this is not an free radical. Hydrogen peroxide is very reactive, but that is also not an free radical. Hypochlorite ions are very reactive. They are also supposed to be not that way a free radical. They are reactive. They are oxidants. They are reactive oxygen species. So what are free radicals? Superoxide anion, as they have only one unshared or unpaired electron. Hydroxyl radical, also the similar way of configuration. So they are highly reactive, and these particular species are known as free radicals. Now, what about the free radicals? Why it is reactive? Why it is so damaging? In order to gain stability, a free radical steals an electron from another compound through a process called oxidation. So it snatches electron from the neighboring compound. The second compound becomes unstable and responds by stealing an electron from another compound, resulting in a chain reaction. So in this example here, the hydroxyl radical snatching an electron from the carbon-centered radical, and carbon-centered radical becomes a free radical, which snatches another electron from the oxygen, so that peroxide radicals are formed. So this way, free radical causes chain reaction, and that's why it is a highly reactive chemicals. So now here is a list of different reactive oxygen species and reactive nitrogen species, and some of them are free radicals. Like here, free radicals are hydroxyl radical, superoxide radical, but hydrogen peroxide, peroxyl, or cygnate oxygen, they are actually, so to say, not free radicals, but they are all reactive oxygen species. Likewise, nitric oxide is definitely a free radical, as they have one unshared unpaired electron. But there are so many other nitrogen species which are not free radicals. They are reactive nitrogen species. They are highly reactive, but not like the free radicals ones. Next, please. Now, how free radicals are generated in our body? Free radicals is definitely generated in our body by external effect as well as by the internal cellular reactions. The different biochemical reactions give rise to free radicals, and also the effect of the external environment also creates a lot of free radical within our cells, out of which very important is cigarette smoking. Cigarette smoke gives us a lot of free radicals through our lungs, and that is very important to learn that why cigarette is so much harmful, so much injurious. Actually, nicotine, the leaf from which the cigarette is produced, is dried in the sun. So during that period, the lot of free radicals generated as because they are exposed to some ultraviolet radiation of the sun, and these ultimately go inside our body when we take the cigarette smoke and causes lot of damage to our tissues. So likewise, air pollution. The different carbon-centered radicals are definitely taken in from the air pollution, the combustion of the carbon compounds, petrols, diesels, etc. Radiation is very important for position. Nowadays, we don't have not only the aerial radiation, but also radiation from different gadgets. Even your cell phone gives rise to a lot of radiation. Television, at least you should have 20 feet distance from the television if you have to avoid the radiation. Pesticides. Nowadays, no food is free of pesticides. Agriculture, food is free of pesticides. So pesticides also generate a lot of free radicals. Different chemotherapy drugs, like anti-malarial drugs, anti-leprosy drugs, they are actually oxidants. They produce free radicals within our body. If they are over used, this can cause a lot of damage, and some sensitive persons, they call some toxic effects. Polyunsaturated fats. Here, polyunsaturated fats means those fats which have lot of double bonds within their chain. And these polyunsaturated fats actually is very liable to damage by generation of the free radicals. Because of the double bond, if there is any free radical generated, the double bond is broken immediately, and they are a source of the polyunsaturated, source of the free radicals mediated damage. If a saturated fatty fats are there, it can actually quench the free radical chain reaction. That you will understand that during the lipid peroxidation reactions, it will be discussed. High altitude, definitely where the frequent flyers or they receive lot of gamma radiation. Now exercise do causes generate some free radicals. Now limited exercise or reasonable exercise is beneficial for our body because that will induce our immune system, that induce our antioxidants. But if we do some over exercise, that is detrimental because that causes a lot of free radicals generated at a time. So all these are causes of generation of free radicals. But as I have told you, the most important aspect of generation of free radicals in our body is the different biochemical reaction, out of which the biggest source of free radical is our own body oxidative phosphorylation. Oxidative phosphorylation you have studied, where reducing equivalents are ultimately oxidized through the respiratory chain. So, to form one H2O, oxygen are ultimately neutralized. So, four hydrogen protons are required to neutralize one oxygen molecule. Now, during the transport of protons through the membrane, the 4 hydrogen and 1 oxygen molecule, or 1 atom and 2 hydrogen molecule, they don't go always simultaneously to be neutralized as H2O. Sometimes or most of the times it goes in a stepwise pattern. Suppose one oxygen first receives one electron, which is also transported from one end to another. Then this becomes a superoxide radical, as you are saying here. Now superoxide radical is acted on by an enzyme superoxide dismutase, which tries to neutralize that to a relatively less reactive molecule H2O2, hydrogen peroxide, by taking another 2 proton plus one another electron. This H2O2 is again, if receives 1 electron and 1 H plus proton, that will be converted to hydroxyl radical, and this ultimately receives further electron and hydrogen proton to give rise to H2O. So stepwise electrification and stepwise receive of these electrons leads to generate different free radicals like hydroxyl radical, superoxide radical. And most of the cases, though the electron are the oxygen simultaneously electron goes with the hydrogen atom, but 5% of the electron are ultimately transported by this stepwise pattern. So, since the cell developed a system of generation of ATPs, more amount of ATPs from one molecule of glucose through the respiratory chain, it has to utilize the mitochondria. It has to utilize the oxygen, oxidative pathways of utilization of glucose. Since that time, whenever oxygen was used as a final electron acceptor, the cells also had a risk of developing some reactive species, since of developing some free radicals, and that's why we have also developed some antioxidant system that we'll see later on. Now there are some other free radical generating reactions which are very common, out of which I have already told you the leaks in the electron transport chain in mitochondria during normal oxidation of foodstuffs, that is the main source But another very important source is the NADPH oxidase. In the inflammatory cells, in the neutrophils, macrophages, this respiratory burst in order to kill the bacteria or virus, pathogens, they generate free radicals. How? The NADPH is formed from the hexose monophosphate pathway. It is required to by the cell in this system and acted on by the NADPH oxidase to form superoxide. This superoxide is needed for the killing purpose of the bacteria as it is converted immediately by SOD to H2O2, and this H2O2 is acted on by the myeloperoxidase enzyme, and along the help of the chloride, it converted to hypochlorous acid, which is highly toxic for the pathogens or bacteria. And so bacterial killing purpose, these are the very important phenomena happens within the inflammatory cells. Now there are some reactions need to be addressed. One is iron catalyzed reactions. Iron catalyzed reactions may be one Fenton reaction. A Fenton reaction, the ferrous iron plus H2O2 is converted to ferric iron plus hydroxide radical. This is Fenton reaction. This always happens whenever iron get a chance to be exposed, maybe within the heme or maybe in the peritoneum. So whenever there is any hemoglobinopathies, the iron is never caged properly, and so there is always chance of causing free radical damage. In case of HbA1c, you know what is HbA1c? HbA1c is the glycated hemoglobin which happens due to the attachment of glucose with the hemoglobin molecule, and as proportional to the concentration of the glucose molecule over the last 2 months, it gives reflection what is the amount of the glycosylated hemoglobin. Normally it is up to 6%, that you know. Now, one of our Bengali scientists, who was my colleague, Dr. Manoj Kaur, first uncovered that this HbA1c gives rise to free radical mediated Fenton's reaction. So HbA1c is a source of free radical generation within the vasculature. That is a very unique finding, and we find out that so oxidative stress, diabetes is associated. That has been established, but one of the sources is the generation of the HbA1c, and by which the Fenton reactions and generation of the hydroxyl radical within the vasculature. Likewise, the Haber-Oasis reaction is also there. Now, lot of UV ray act on by our cellular water gives rise to, if we are exposed to the UV ray X-ray, then that will be causing free radical generation. Now, one of the important free radical is nitric oxide that I have told you, and this is generated within our cell by the reaction of oxygen with L-arginine, and L-arginine is converted to nitric oxide plus citrulline by a group of enzymes known as nitric oxide synthase. There are different types of nitric oxide synthase, and it has been studied very interestingly. If nitric oxide synthase has certain changes or polymorphism or some defective enzymes that converts that superoxide with the help of L-arginine to oxygen with the help of L-arginine to superoxide rather than nitric oxide. So that is another findings which always can generate some of the unwanted free radicals. Though nitric oxide is very beneficial, sometimes that we will see most of the cases nitric oxide is required by the cell, not only as a signal transduction molecule but on many of the cellular functions. So xanthine oxidase, another pathway by which free radicals are generated, lipooxygenase pathway that you will find in the platelets and leukocytes are also very much associated with the generation of the free radicals. You will study in more detail later on, but these are the basic things that you should know. Next, please. Now, how free radicals damage the cells. Now, you have three main compartments. One is the plasma membrane made of mainly lipids. Then there are cell structures mainly constituted by interaction of different proteins, and the nucleic acids contain DNA. So all three major aspects of the cell can be damaged. The cell membrane can be damaged by lipid peroxidation reactions. There may be protein which can be damaged by different protein nitration, cross-linking of the protein, and DNA. There may be a lot of mutation caused by the free radicals. So widespread destruction over a long period contribute to problems. What problems? Cancer, diabetes, atherosclerosis, kidney disease, accelerate aging, and many others. That is why there is where the antioxidants come into play. Most people are not suffered from this disease because they have a proper balance of antioxidant within their body. Next. Now, antioxidants, there are a lot of molecules considered as antioxidants. So what are basically antioxidants? Antioxidants are the compounds which actually can donate an electron to neutralize a free radical. But doing this, it is itself become a oxidant. That we'll see later on, but first we have a list of antioxidants here. Now, body has its own defense system in the form of enzymes. Superoxide dismutase, I have already told you, catalase, glutathione peroxidase, and glutathione reductase. They are all antioxidant enzymes working in our cell, within our cell. There are some vitamins which are also antioxidants. Alpha tocopherol, beta carotene or vitamin A. Alpha tocopherol means vitamin E, and ascorbic acid, vitamin C. Vitamin K can also be considered as antioxidant vitamins. Several small molecules are also working as antioxidants in our body, of which glutathione is very important. Glutathione tripeptide is very, very important. So, along with, we have uric acid and bilirubin. They are small molecules, but their effect may be a little less, but they also contribute considerably in the antioxidant, as antioxidants in our body. Now, there are some naturally occurring antioxidants in the plant pigments, in the flavonoids. Natural iron chelators are also antioxidants, as iron actually, when it is exposed, it is always a source of generation of free radicals. So iron chelators are naturally considered as antioxidants. Synthetic molecules, there are a lot of synthetic molecules now developed as antioxidants. Now, xanthine oxidase, NOS inhibitors, SOD catalase, glutathione peroxidase mimics, they are being produced in the laboratories and they are tried in the different disease condition that we'll understand gradually in the due course. Next please. So first here we see how antioxidant protects ourselves. Taking the example of superoxide ion. Here superoxide anions, see superoxide dismutate is working, taking two superoxide anions here and converting them to one oxygen and another hydrogen peroxide, which hydrogen peroxide is definitely a reactive species, reactive oxygen species, but not reactive as superoxide. So they are rather neutralized by the effect of the superoxide dismutate. There are different types of superoxide dismutate within our cell. That may be membrane bound, that may be cellular. So that we will understand in due course and different disease situations. So how this antioxidant protect ourselves? They actually scavenge ROS or RNS. ROS means reactive oxygen species, RNS reactive nitrogen species. They also prevent their formation. Sometimes it has been seen the antioxidant prevent the generation of the free radicals. Sometimes they upregulate the endogenous defense system. And some case aiding the repair of damage. So the antioxidant mechanisms not only scavenge the ROS or RNS, they also prevent their formation, upregulate endogenous defense system, as well as they aid the repair in damage. So this way, overall, they protect from the free radical mediated damage. Next, please. Now, as I have told you, the antioxidants reactions scavenging free radicals. There are some examples here. SOD I have already told you. Now, catalase is very important. It is one of the peroxidase enzyme actually, which specifically act on the hydrogen peroxide wherever generated is clipped by this enzyme into H2O and O2. G6PD is a very important enzyme in the pathway of hexose monophosphate shunt, which helps us generate NADPH. And NADPH is a very important reducing equivalence, which is actually involved in the reductive biosynthesis. Here, also very important as antioxidant. Now, enzymes like glutathione peroxidase and glutathione reductase, they are involved in free radical mediated reactions. Glutathione plays a very important role. Now, glutathione in the cell usually kept as the reducing state, that is glutathione, reduced glutathione. Now, whenever there is a free radical mediated damage, the reduced glutathione is converted to oxidized glutathione by the interaction of the enzyme glutathione peroxidase. This glutathione peroxidase is a selenium containing enzyme. And then this GSSG has to be further reduced for quenching another free radical mediated damage. So that has to be regenerated, and they regenerate with the help of glutathione reductase enzyme, which requires the NADPH plus H plus. And this NADPH plus H plus is generated through the pentose phosphate pathway. So this is a very important reactions considering the free radical and antioxidant mechanism and keeping the balance properly so that the body functions normally. Next please. Here is an example of some damage in the lipids. Basically, lipid peroxidation reaction. As I have told you earlier, lipids are very prone to develop free radical mediated damage because lipid is bilayer forms the plasma membrane. And plasma membrane is the organelle which makes the environment a cell of the cell where the biological reaction is possible, the life reaction is possible. Now, in this, lipid are very prone to develop free radical mediated damage. Now, in normally physiological situation, free radicals are generated, and a small amount of free radicals which are generated are actually neutralized by the body's own mechanism. So, repeat peroxidation reaction basically there is an initiation phase, there is a propagation phase, and there is a termination phase by the own reactive mechanism. That is balanced control. Here you can go through that PUFA, free radical mediated damage occurs, uh, and that causes generation of lipid radical. This lipid peroxidal radical are formed, and they ultimately propagate to through the chain to some extent, and ultimately the different type of uh, free radicals interacted with each other to be neutralized by themselves. So whenever there is limited or generation of the free radicals, the lipid itself can neutralize the reactions. But in case of larger amount of free radical mediated damage, these self-limiting reactions cannot be stopped. So there is a chance of damage. In case of polyunsaturated fatty acid, more damage is expected, and there is no way of quenching that particular extra electron in those cases. So they will be broken very rapidly. Saturated fatty acids, if it is there, that can donate one electron and make themselves shifted to unsaturated ones and thereby quench. So in the cell membrane, three types of fatty acids are very important: monounsaturated, polyunsaturated, and also the saturated fatty acids. All is required to be present in a ratio of 1:1:1. Anyway, that's another question. You have to learn the lipid bilayer membrane, how they are made up, and what is the advantage and disadvantage of the lipid constituents of the membrane. Now, here in this case, these particular reactions are also sometimes whenever excess, they are added by some of the checked by some of the antioxidants playing a very important role. One of which some of the antioxidants, vitamins, which stays in the lipid phase plays the role initially, like vitamin A and vitamin E. Vitamin A actually very very important because it helps to stop the initiation of the free radical mediated damage. We'll see it in the next slide, please. Now here is the lipid peroxidation reaction, introduction of antioxidants in the lipid phase as well as in the aqueous phase. Just go through this diagram. The lipid free radical mediated damage gives rise to PUPA change into peroxide radical, and this peroxide radical formation can be stopped initially by vitamin A. But when it is already produced, the reaction is continuing through the chain, then it is taken care of by the vitamin E or reduced tocopherol. You take reduced tocopherol, neutralizing that free radical into PUFA, and they become themselves an oxidized ones. And the PUFA, that is the peroxide radical, is neutralized with the help of SOD and catalase system in our body. In the cell, there is a very important and vital reactions carried out by playing role in the form of vitamin C. What does vitamin C? Vitamin C helps or works in conjunction with the vitamin E because this oxidized tocopherol need to be again reduced with the help of vitamin C or reduced ascorbic acid to oxidized ascorbic acid, ascorbate, and that is excreted. And so we have to take vitamin C adequately in our diet so that every time we can be able to quench the free radical mediated reactions. Now, another one I have already told you, GSH plays very important role to neutralize the H2O2 to normal ones, and GSH is actually reduced glutathione by the process becomes oxidized with the help of glutathione peroxidase, and this oxidized glutathione again regenerated into reduced glutathione with the help of glutathione reductase. So the NADPH is playing very important role here, which is generated by the hexose monophosphate SUN, and G6PD is a very important enzyme in this aspect. Next, please. So I think now you have already understood what is oxidative stress. Stress means there is a disbalance. A disbalance or disturbance in the balance between reactive species or free radicals and the antioxidants in any system, any tissues give rise to the oxidative stress in that particular tissue, organs, or the cells. Under normal condition, cells are able to balance the production of oxidants and antioxidants, and they are perfectly playing very important role because we require to generate oxidants. That is, oxidants are very much important for our cellular biochemistry. They will be definitely generated. Some antioxidants oxidants are also helpful for us, and antioxidants is there right from the beginning to take care of the excess of antioxidants. If that balance is lost, the oxidative stress occurs. So when there is imbalance in our cells due to either an increase in the free radicals or reactive species or a decrease in the amount of the antioxidants, then this balance is disturbed and there is oxidative stress, and this stress over the years gives rise to certain disease. So that is the balance I have already told you. So disturbance of the balance in favor of free radicals gives rise to the oxidative stress. Next, please. Now, how we can measure in your profession? You will have to encounter a lot of disease associated with different free radical mediated damage. But you should know how to assess a patient whether he is suffering from any oxidative stress or not. Though these type of tests are not till yet normally introduced in our laboratories in routine practice, but research workers are already developed certain methods by which we can identify whether the oxidative stress is there in that particular situation or not. Though the investigations are done in different laboratories, but not in the clinical laboratories routinely. The lipid peroxidation products can be measured like thiobarbituric acid derivatives like malondialdehyde. The lipid peroxidation products can be measured, gives rise an idea what amount of peroxidation is going on, what amount of oxidative stress is going on. Hydroperoxides can also be measured. DNA hydroxylation products can also be measured. The protein hydroxylation products like tyrosine nitration products can also be measured that will give rise to an oxidative markers. Now, free radical detection can also be done by single photon counting, by chemiluminescence, by fluorescent probe, and by electron parametric resonance spectroscopy. The assay of antioxidants in the body fluids or tissues are done mainly by estimation of different enzymes, antioxidant enzymes like SOD, catalase, and glutathione. And also we can estimate vitamins like vitamin C, vitamin A, vitamin E, etc. Those I have told you, these are not routinely practiced till nowadays, but you should know this oxidative stress can also be assessed during your study. In a higher study, you can have certain research works with some disease where you will be maybe needed to estimate the exact status of the cell. So you can take any of the uh, methods here. But what is more important, that why you will be trying to know about the status. Most of the cases we don't know, but we still practice therapy with antioxidants. So whether antioxidants should be used or not, whether the antioxidants can be introduced in this patient or not, whether antioxidants is required by the patient or not, that has to be ascertained. That is the problem with the modern day therapeutics, because in the treatment of human disease, lot of antioxidants are not being used, and also dietary antioxidants in the disease preventions are advised. But whether you at all require, whether you have shortage of antioxidants or whether you don't, that you have to first look after. Because battery of antioxidant drugs are available in the market, they are very often being prescribed by the doctors. I'm sorry to say, before understanding whether the oxidative stress is due to deficiency of antioxidants, but they are as prescribing antioxidants, because I have seen in the patients suffering from oral submucous fibrosis are often advised a lot of antioxidant medications. So you have to assess that if it is possible. Now, continuous research, because continuous research over the 25 years have contradicted informations regarding the role of the antioxidants, regarding the role of a particular disease situation. Antioxidants can either offer protective effects or even worsen the occurring damage. So you don't know whether it is going to damage further. Now, one study, vitamin C was seen when taken less as per RDA recommendations. The study subjects associated with increased free radical mediated damage to DNA. Paradoxically, the same was observed with high dosage of vitamin C. Why? That is very important to understand, because all antioxidants are potentially pro-oxidants. Why? Antioxidants, I've told you, they act by scavenging. The resulting antioxidant derived radicals themselves become pro-oxidants and they can again damage. So before we advise or advocate any antioxidant, we should always know whether they require antioxidants or not, whether this patient actually suffering from oxidative stress or not. So oxidative injury may involve damage not to lipids but proteins or some other things. So inhibiting lipid peroxidation may not protect the tissue. So inadvertent use of the antioxidant should not be there. Though you are students nowadays, but you should develop this concept at least, whether or not the effectiveness of the antioxidants in treating disease is beneficial, only to those with a decreased oxidant status. If you have a decreased status, then only you can consider certain things. So please try to develop the concept right now. Try to know what are antioxidants, what are oxidants, how they are basically generated in our cell, how they are generated by the effect of the external environment, what are the different way of mechanism is there which generate a lot of uh, defense system by antioxidant present in our body, and if there is any problem, any disbalance, there may be some disease associated uh, with the antioxidant system, with this free radical mediated system. So that is important to understand whether or not the oxidative stress is significant contributor to the disease pathology. A common interpretation at the time of study. But over the course of the study, there might be other important pathological causes. So antioxidant is not always the answer. So please bear with it and keep it in your mind. Now, some of the oxidants are beneficial in our body. Not all free radicals are bad boys. They are also very helpful. Some ROS, RNAs have useful role in vivo. Could the proposed antioxidant interfere with this? That you have to always understand. Now, nitric oxide is an excellent molecule which is a signal transduction molecule, and there is no tissue where no cell, no biological biochemical reactions where important biochemical reaction where nitric oxide does not play any important role in the nerve signals, in the blood vessels dilatation, in apoptosis, in so many things. And hydroxyl radical is very important. You require to generate it for the defense purpose for killing the bacteria. So if you have more or excess of antioxidant, are they going to interfere with these beneficial ones? So that you have to again consider and know. So better we should be checking or access the patient to confirm this before administering antioxidants as a treatment method. So this is a concept you should develop. Not always you will be able to measure the things, but you should try to evaluate the patient in that way. And lot of things you have to learn during the course of medical science, but antioxidant and free radical biochemistry is always associated in the disease pathology, the etiology of many disease, and recently it is considered to be on the modality of drug. So please try to develop this concept. If you have any doubts, we'll be able to answer your doubts, you'll be able to address your doubts during the interactive sessions. Thank you very much.